All right, bio buddies. Assuming you've seen the presentation on passive transport and diffusion, you are now ready to explore the wonders of osmosis. All right, so the definition of osmosis is just the process by which water diffuses across a membrane. Now, we're still talking about diffusion here. So we're still talking about going from high concentration to low concentration, and we're still talking about a situation where the cell doesn't have to expend any energy. Why do we give the diffusion of water a special name? Well, remember that cell membrane is selectively permeable. And if we're looking at various solute molecules, some can get across, some can't, depending upon whether or not they have a charge, whether or not they're large, they're small, whatever chemical characteristics they have. But remember that water can always get across cell membranes because you have those special channel proteins called aquaporins. And since those aquaporins are always there, the cell is always going to be, or the cell membrane is always going to be permeable to water, regardless of what else can move. So we're going to focus a lot on water movement here. Okay, we're going to give you 3 different situations, and you're going to have to analyze what's happening in terms of water movement in each of the situations. Remember that water is always going to go from higher concentration to lower concentration. Okay? All right. Now the first type of solution you could put a cell in is an isotonic solution. Iso means same. So if we're looking at isotonic, we're looking at the concentration of solutes, and we're always talking about solutes here. The concentration of solutes is the same in the solution as it is in the cell. So if we went ahead and put a hypothetical cell in solution, we would have the same concentration, in this case, 10% solutes inside the cell, 10% solutes in the solution. Thus, it's isotonic. But here's the kicker. If you have 10% solutes in the cell, that means the other 90% is water. And same thing in the solution. If you've got 10% solutes, well, you got 90% water. We don't know if the solutes can get across the cell membrane because we haven't identified what solute we're talking about. But we know that water can get across the cell membrane. And since you have the same concentration of water on this side as you do on that side, yeah, sure, water molecules may leave the cell, but chances are just as many water molecules are going to enter the cell. So theoretically, in an isotonic solution, the cell should be in equilibrium with respect to water, and you end up with no net movement of water either going inside or outside the cell. Now, isotonic isn't all that exciting because nothing seems to be happening. But these next two, you're going to see something happening for sure. Hypertonic. Hyper means more than, greater than. So if we're talking about a hypertonic solution, remember you're always going to be looking at the solution. That means the concentration of solutes is greater than that in the cell. In this case, I got 20% solutes on the outside in the solution, 10% solutes on the inside in the cell. All right, well, again, you don't know if those solutes can move across the membrane, but you do know that water can move across the membrane. So you flip it. If you got 20% solutes, that must mean you have 80% water outside the cell. If you have 10% solutes in the cell, that means you have 90% water inside the cell. Remember, water is always going to move from areas of high concentration to areas of low concentration. So in this case, hey, 90 is greater than 80. Water should leave the cell. Right? And those are the questions that you always want to ask. What is the concentration of water in the solution? what is the concentration of water in the cell? And you have to realize that water is always going to move from areas of high concentration to low concentration. So in a hypertonic solution, the cell is actually going to lose water. And we'll talk about some applications of that in class. Well, hypotonic. Hypotonic. Hypo means less than or lower than. So again, we're talking about concentration of solutes, but concentration of solutes in the solution. So in our solution, we can change our numbers a little bit, use the same cell, 10% dissolved solutes in the cell, but here the concentration of dissolved solutes is less in the solution. This makes it hypotonic. Again, you can't tell what the solute is going to be doing unless you identify the solute. It may or may not be able to get across the membrane. But you know that water will always get across the membrane. So, if you have 5% dissolved solute in solution, that means you must have 95% water. Since 95% water is greater than 90% water inside the cell, water is naturally going to move from areas of high concentration to areas of low concentration and enter the cell. So in hypotonic situations, the cell is going to gain water. Now, since the cell's cytoplasm is normally made up mostly of water, you're going to have a lot of internal water pressure inside a cell. And this is actually kind of a good thing because it gives the cell kind of firmness, it gives it kind of an internal support. Right? So if we're looking at, say, this plant cell's got this huge, centrally located, water-filled vacuole, and in that vacuole, if it's full, has a certain amount of internal water pressure. We call that osmotic pressure or turgor. Okay? Now, if that vacuole is filled, then the plants are well supported from the inside out. Each and every individual cell is well supported, so the plant is well supported. However, if we remove some of that water, if there's not as much internal pressure there in that vacuole, that cell is going to collapse a little bit. And if each individual cell collapses, the whole plant is going to collapse, and you'll have this kind of droopy, wilted effect. So turgor pressure is actually a decent thing for a cell because it keeps the cell from collapsing. Well, you can have either too much turgor pressure or too little turgor pressure, and both of them are going to affect the cell profoundly. So if we looked at, say, a normal human red blood cell, it would look something like this, right? And they look kind of like a disc with that's been squeezed in the middle. A normal plant cell, well supported down here, right? And both of these might be in isotonic situations. If, however, we were to go ahead and put either of these cells in a hypotonic situation. Remember, in a hypotonic situation, you're going to have a less amount of solute in the solution that they're in, but that means there's going to be a greater amount of water. So water is going to enter the cell. Well, if the turgor pressure becomes too great in an animal cell, the cell might undergo cytolysis. Cytolysis is when the cell actually goes ahead and bursts because of, well, basically it's just overinflated. It's like taking a balloon and pumping in more and more air, or in this case water, until it just bursts. Cytolysis is unlikely to happen in a plant cell because, remember, plant cells have that cell wall that surrounds them. So imagine taking your balloon or your water balloon and inflating it, but having the balloon encased in a box. The walls of the box keep the balloon from popping. So, the opposite can occur if we put the cell in a hypertonic solution. If we put it in a hypertonic solution, you end up with the cell losing water. Remember, in a hypertonic solution, the amount of solutes is greater in solution than it is inside the cell. But that means there's less water in solution, or less concentration of water in solution than there is in the cell. So water will move from high concentration inside the cell to a lower concentration outside the cell. The cell will lose water, and if you happen to be an animal cell like this, again, little red blood cell, it's going to shrivel up, shrink, and collapse. Once again, a plant cell might have the same thing going on. Water will drain out of its vacuole as it loses water. It might even have its cell membrane peel off the cell wall, and you'll have this internal collapse. But the cell walls will still remain on the outside. In the case of animal cells, this collapse we call crenation. In plant cells, they call it plasmolysis. Now, if the cell happens to be in a hypertonic or hypotonic situation, that doesn't necessarily mean it's going to die. It could, but protists, single-celled organisms, oftentimes live in fresh water where they're in a naturally hypotonic situation. And normally they would just take in water, and burst. They don't have a cell wall like a plant. But they do have an organelle called a contractile vacuole. And this one here is an empty contractile vacuole. Over here is one that's full. Well, the contractile vacuole is there to actually go ahead and soak up water from the cytoplasm, and then every once in a while it will fill completely and then squeeze out the excess water, almost like it's wringing out a sponge. Now, it has to do this 24/7. Every second of every day throughout this cell's lifespan, it has to go ahead and take up the excess water from the cytoplasm and squeeze it out. If it doesn't, it's going to explode. Now, this does take some energy, but hey, if the alternative is explosion, then you might want to just go ahead and use up the energy. Now, plant cells don't have to worry about cytolysis because, like I say, they got that cell membrane. But hey, there's got to be other ways of doing this. All kinds of organisms live in hypertonic and hypotonic situations, and not all of them have contractile vacuoles. Well, you can move substances against the flow of osmosis and diffusion, against the concentration gradient, using energy. And this involves something called active transport. Remember, in diffusion, it's a lot like taking that basketball, leaving it unsupported, and you don't have to have any energy at all, any energy input to get it from that high point to the low point. And molecules will naturally go from high concentration to low concentration. But how could you get that ball from that low point to the high point? Well, unless you change the ramp, you have to actually put some energy into this to move it, to push it back uphill. And that's kind of sort of what active transport is all about. And we'll talk about that in our next video. So, something to look forward to. Anyway, your next task is to go to our next slide. And here we got some review questions, which would be probably pretty darn similar to what you might find on a quiz. So make sure if you have problems answering these questions, you see me, let's talk about it so that you'll have no problem with the quiz. Anyway, have a great one. Till next time. See ya.